Hey everyone. I figured the best way maybe to talk through this EOC practice test would be to actually talk through it versus just, posting an answer key online. That being said, I also will post just the answer key online in the form of what looks like kind of like this, where it's just bubbled in. But what I'm going to do in this screencast is talk you through what the answers were supposed to be and explain hopefully why those answers were what they were. All right. So feel free to, if this isn't helpful for you, certainly you don't have to watch the entire thing, or you can try to skip around to find explanations to questions that you actually maybe had questions about or were confused by. All right. So I'm going to start here with the sample question, which I'm assuming most of us also started with. And let me see if I can make this here a little bit smaller so it fits a little bit better. So when we look at, let me change that again. We look at a question like this. This is again, it's a sample question, so I don't have an answer for this in the back of the book, like in my teacher's edition, right? But we still can work through this, and it is still good review for the content that they're presenting, which is about biotechnology and gel electrophoresis. So the scenario is we're looking at hair samples. We have those samples that have been obviously digested with restriction enzymes and cut up. And then they've run a gel electrophoresis for each of the 5 samples. And one of those is what we're calling the unknown sample. So what we're trying to identify based on the question that they're asking, which sample is from the person who is most closely related to the person who was selected for the unknown sample. And so what we want to do with this is to compare my samples here, 1, 2, 3, and 4, to my unknown sample here on the very, very left. And what I'm looking for is similarities in what we call these banding patterns. So in general, the more bands that they share in common, meaning the same position kind of across, the more likely those things are going to be related or potentially from the same individual. So if you remember about gel electrophoresis, what these bands actually represent are fragments of DNA, and they travel from largest to smallest in size. So my very smallest pieces of DNA are down here at the bottom, and the longest pieces of DNA, after being cut with restriction enzymes, are going to sit up here at the top. Okay, so for any DNA, like for example for me or for you, if you were to analyze any DNA sample from one particular living thing, you're going to get the same fragmenting pattern every single time. So in this example, the one that kind of sticks out to me that looks most similar is going to be sample 1. So these 2 bands at the very top are the same. You've got this one here, the 3rd band down. This one here is in line, and then this one here at the bottom is in line. So it appears that sample 1, for all of those bands except for this one here in the middle, is pretty well aligned with unknown sample. Sample 2, on the other hand, doesn't look very similar. It's got some bands in places where we don't have bands in the unknown. Same for sample 3, which is, a little bit more consistent with the unknown. But sample 4 is just kind of all over the place, all right? So again, what you're looking for these questions about most closely related is going to be those that share the same types of patterns, right? The DNA fragments that we see. All right, so number 1 was mostly just a recall question. It said, which change in DNA will cause a heritable germline mutation? And what that really is referring to, and part of the issue I think with this test is that the vocab becomes a little bit tricky. So when we say heritable, that means that it's inherited. That means it's passed from parents to offspring. And so what that should make you think of in your mind is going to be, oh, it's got to be genetic. It has to be DNA. It has to be RNA, something like that. Germline refers to that line of cells, that group of cells that we talked about back in chapter 11 and 12, which are used to produce gametes. So germline mutations refer to mutations that would be happening in your germ cells. And those germ cells are what over time develop into sperm and egg, okay, aka gametes, aka sex cells, okay? So if we're thinking about a germline mutation, we want to see a change happening in gamete or gamete-related cells. So the first answer option here says we have a mutation to the DNA in bone cells. And bone cells are somatic. Remember, that means that they're found in the body, not in the cell groups that will eventually be donated to offspring. So this one doesn't work because it's a somatic type of cell. The next one down, B, says we have a mutation of DNA in a skin cell, also a somatic cell, also not passed down from parents to offspring. C, we have an error that occurs in a sperm-producing cell. So sperm, we know, has to do with reproduction. So that one looks to be pretty good. And then D, an error occurs during transcription of the DNA in a liver cell. And liver cells also, like bone cells and skin cells, are also considered somatic. And just keep in mind, anything that's somatic, in other words, anything in the body that is not gamete specific, that is not sperm or egg, or going to develop into those, like the germ cells that I mentioned, any of those will not get passed down. It's only those germ cell lines and the gametes that are produced from those that we end up passing down to our offspring. So C is your correct answer for this one. Okay. And I've got a printed test here, so if you see me looking off to the side, I'm just flipping through because I made some notes in it also. Question 2, a student is investigating how substrate concentration affects the rate of an enzyme catalyzed reaction. Which 3 variables should be kept constant in the setup of the student's investigation? So we're looking at really a couple of things here: enzyme activity and also scientific method. How do we set up a good experiment? When we talk about variables, those are things that change in an experiment, but if I want to keep these things constant, that's what we refer to as constants. And so remember, in any experiment, we want to have a lot of constants because that makes our experiment a little bit more accurate and valid. And so we're looking for a number of those. But what we cannot keep constant is the thing that we're measuring and also the thing that we're testing, right? So what we call our dependent variable, what we're measuring, and our independent variable, what we're testing. And so if I look at the setup, we want to know how substrate concentration affects the rate of an enzyme catalyzed reaction. So substrates, if you remember, is just a fancy scientific word for reactant. So if we're looking at the amount of reactant and how that impacts how an enzyme functions, then we don't want to keep that amount of reactant the same, right? The amount of reactant possibly, like, it can't be my constant because I'm looking at how the amount of that changes the products that are being made. And so the amount of substrate there are, which you see listed here as an answer option, cannot work in this example because it is the one thing that does get changed, right? It's the independent variable. It's the purpose for doing the whole experiment. So that is not a constant. The other thing that cannot be a constant is S, the amount of product, because that's how we're measuring the activity of the enzyme, right? Enzymes, remember, are biological catalysts, so their jobs are going to be to catalyze and speed up chemical reactions by lowering the activation energy required to do the reaction. And so as a result of that, if their job is to kind of push a chemical reaction on, well, then the product is a way that we can measure how well they're doing that, right? So the amount of product that's made by these enzymes through their activity is what we measure in this type of experiment. So the amount of product is also not a constant. That leaves us with 3 things that could be constants, and those are your 3 correct answers for this. M, which is temperature of the reaction, and that we want to keep the same because we're not trying to investigate that part of this. P, the amount of enzymes, so we don't want to change that if we're looking at the substrate or the reactant specifically. And then T, the pH of the reaction, because we know pH changes can alter structure and function of enzymes, and we don't want that here, okay? So again, answers there, M, P, and T. Number 3, we have an island that's part of an island chain that's recently been open for tourism. All right, that's a scenario. So there haven't been new human interactions happening there. And so the question is about succession. And if you remember, succession is a piece of or a period of changes that occurs in an ecosystem due to something. So it's either going to be replacement of a damaged ecosystem, which is what we call secondary succession, or it's going to be the growth of an ecosystem in a community in a place where none existed before, where we're essentially starting with bare rock. And that's called primary succession. In this example, since we're starting with an island that's been there, I mean, we assume it's been there because we're saying people were not allowed on the island. So our assumption would be that there are probably other living things present and that those living things were being protected. If there is something there, what that indicates is that whatever is there is going to continue kind of growing in that same pattern, except for now, if we're going to bring in humans, humans tend to have negative impacts on their environment. They're not great in terms of conservation. They tend to build things, which destroys habitats. And so as a result of that, what we tend to usually see with habitats in terms of human interaction is we see a decrease in biodiversity. So your answer here is going to be C. Biodiversity decreases as the number of species increases. The rest of these kind of point to either the lack of human interaction, which would be D, which is where we actually have more species that move into the area that increases biodiversity. That might be true if there were not humans present at all. A here is pointing to what we call primary succession, which would not happen because we don't need lichen as part of this. We already have things living on the island. And then B is saying kind of something is backwards, which is that we have these very mature trees and forests that somehow create a pioneer community. And a pioneer community is the very first step of primary succession, where truly there's nothing existing in a place. Like it's bare rock, and we have lichen that start to grow as the pioneer species, but we don't have even small plants growing yet. Okay, so B just doesn't work because it's just kind of flipped around. It's kind of oddly worded there. Okay, so again, answer there is C. Number 4, a cell model is shown. Students were instructed to review the types and number of structures that are present in this cell model. Which 3 cell types can be represented by the model? So you can see they're labeling for you very nicely here, lots of organelles. A couple things to point out. This cell is spherical. It is round. This cell also seems to lack chloroplast, but it has mitochondria. It also has lysosomes, and it also has a nucleus and other membrane-bound organelles. Notice it's not box-like. Notice it is not surrounded by a cell wall. And so that's going to help us to rule out some of these answer options here at the bottom. So if we think about cells that are spherical, those tend to be things like animals. Those tend to be things like some protists, and even some bacteria can be spherical. But the difference is that bacteria, remember, don't have all these really complex membrane-bound organelles. So our bacterial cell isn't going to work, because if this is a bacterial cell, you would probably see a cell membrane. It'd probably be a cell that's kind of oval in shape. You'd have a cell wall outside of that, and then you'd really just have ribosomes and some wound up DNA sitting there in the cytoplasm, and that's going to be it. Here we see things that are much more complicated. So it's definitely a eukaryote, which means bacteria don't count, right? Those are prokaryotes. So among the other options, we know that spherical eukaryotic cells, again, can be animals, and humans are animals. So M is a good answer option. That's one of your 3. That can be a human stem cell. P says plant xylem cell, and again, a couple of problems with this. I don't have a box-like cell, which we would expect for plants. I don't have chloroplast, which we would also expect for plants. And I do have lysosomes, which we would expect to see here in an animal cell. And so for that reason, P does not work. Okay, it's not an answer for this one. S, however, does. That's an animal. Sea urchins are animals. And an embryonic cell would have structures like this, given that it's an animal in that type of organism. And then for T, I have mammalian white blood cell. And mammalian refers to the fact that it's coming from a mammal, which is exactly like us, right? Warm-blooded, live birth, those types of things. And so mammalian white blood cell could certainly look something like what you see right here. So again, your answers for these, the correct ones, M, S, and T. Okay. Question 5, we have 2 dogs, a male dog and a female dog. The question is about reproduction, and we know that animals in general kind of prefer to reproduce sexually because it adds some variation to a population. That's going to be super helpful when it comes to survival in an environment. So we know that sexual reproduction does a few things. It creates uniqueness in offspring, and obviously my male and my female in this picture are not exactly the same. So answer option A doesn't work, because we know that they were likely produced through sexual reproduction, and those 2 animals are not identical. So A doesn't work because sexual reproduction creates unique offspring. B, gametes from both dogs are needed to maintain the correct number of chromosomes. Well, that kind of makes sense, because you have a male that's going to give this number and a female that's going to give this number, and then those 2 combine to create a diploid organism as the offspring. So that one works. What about C? It says phenotypic variation will occur in the offspring as a result of mitosis in the gametes. Gametes are produced through meiosis, not mitosis. So that one doesn't work, simply because of that word mitosis. And then D, binary fission is required for sexual and asexual reproduction in dogs. First of all, dogs don't do asexual reproduction ever. And second, binary fission only occurs in prokaryotes, meaning only occurs in bacteria, not in eukaryotes. All right. So your answer for this one is B. All right. Question 6, we're looking at the carbon cycle. And the question here is, which change would lead to higher CO2 levels in the atmosphere? And let me zoom out a little bit of this one. So higher CO2 levels, we have, again, we're just choosing one of these on this. So M says an increase in photosynthesis. If we think about that, think about your reactants, right? Plants absorb CO2 that we breathe out. And so if we're talking about doing more photosynthesis, that means we're absorbing more CO2, or plants are, and that won't lead to more CO2 in the atmosphere. So M doesn't work, because if we're increasing photosynthesis, we're actually pulling more out of the atmosphere. It's decreasing in the atmosphere. P, a decrease in animal respiration. So respiration is something that all living organisms do if they're eukaryotes, and even if they're prokaryotes, they have anaerobic forms of what we do in our cells. Respiration, if you remember, is the opposite of photosynthesis. It pushes out CO2. So if we're doing less respiration, right, we're going to be pushing out less CO2, which is not going to lead to higher CO2 levels. All right, it's going to do the opposite again. It's going to decrease the amount. R, an increase in the burning of fossil fuels. So if we add an increase in burning of fossil fuels, which is happening right here, well, we are going to release pollutants and CO2 up in the atmosphere. So that should actually increase CO2 levels in the atmosphere. And then S, a decrease in the destruction of forests. If we decrease how much we're destroying forest, this is kind of almost like a neutral effect, because the plants aren't going to be respiring and doing respiration to push out CO2, but they're also not going to be bringing it in because they're no longer present. And so it's going to be more of a, like I said, a neutral type of effect. So your best answer option for this is going to be R, an increase in burning fossil fuels. All right, 7 through 10, I'm not going to read through this whole one to save us some time, but certainly if you did not read it, I would definitely recommend doing it. Essentially, we have a green sea slug, and it lives in the oceans, and it's going to use chloroplast from an algae that it eats to help it produce its own food. So sea slugs are animals. They don't normally have the ability to photosynthesize, but when they incorporate and consume these algae, they're going to store their chloroplast in a way that allows them to produce their own energy. So we've got a graph here that results from this, and I think we've got several questions that follow in this scenario. So 7 says, which statement best explains how the green sea slug is able to produce its own food? And so when we think about this one, there's a couple things we want to keep in mind. We know that the green sea slug, based on the information that it started with and gave me here, we know that they have to use chloroplast, right? And we also know that chloroplasts are responsible for photosynthesis. So if you look at A as the first answer option here, the slug is able to convert the oxygen needed for cellular respiration into glucose in the chloroplast. Now that is respiration, which we all know or should know does not happen in the chloroplast. Respiration happens in the mitochondrion. So A does not work. B says the slug is able to store algae within its body and use the glucose produced by the algae for cellular respiration. Well, it doesn't seem to do that, because in the intro materials, the reading of a couple pages ago, it emphasized that they're consuming the algae for energy, and then they just incorporate those chloroplasts. They're not storing the algae in their body. They're going to consume them as a food source. Okay, so that one doesn't work also. C says the slug is able to use a chloroplast to make glucose that it then uses for cellular respiration, and that seems to be written fairly well and accurately, right? Chloroplasts through photosynthesis make glucose, and then glucose can be used in respiration to make ATP energy. D, the slug is able to perform cellular respiration to provide the carbon dioxide needed for photosynthesis. And that one is not so bad, I should say. So we have cellular respiration that we know will make CO2, but that's not the missing piece here for these green sea slugs. The reason why these slugs are able to survive using photosynthesis is not because they do or they do not have CO2. It's because they have to have those chloroplasts. So you want to find the answer that really focuses on the addition of those chloroplast organelles. So C is your best answer for this one. Number 8, which property would give the green sea slug capability to incorporate the genes of the algae into its own DNA? And so with this one, we're looking at exactly, what do living things share in common in terms of genetic information? So there are some things that we just don't know based on the scenario that they gave us. So M says a green sea slug has the ability to replicate the algal DNA. Sure, perhaps, right? But that's not why it can put DNA into its chromosomes, right? It doesn't have to be able to replicate. It just has to be able to kind of stick it there, okay? So, okay answer, but maybe not the best. We'll see what the other ones have. P says the green sea slug has the ability to change its DNA to match the algal DNA. And that's not going to be the case because we don't want to change the original DNA. They simply want to add the DNA from the algae to be able to make those chloroplasts and make them function properly. R, they both have the same number of chromosomes in their DNA. That truly doesn't matter. Remember, the number of chromosomes has nothing to do with how complex something is or what types of genes they may hold. So that one's not going to work also. And then S, they both use the same nucleotides in their DNA. And that is a pretty vague statement, but it's just saying that, hey, we all use A's, T's, G's, and C's, and that is certainly an accurate statement to make. So actually, your best answer option here is going to be S. Okay, they both use the same nucleotides. Number 9, we're looking at a statement that was made here in this case. In the investigation of oxygen dynamics, photosynthesis is reported after 7 months in culture, even when there is no oxygen being produced. And so the question then is, what's the reasoning behind this? So in other words, they're producing oxygen, right? Anytime we do photosynthesis, a product that we know we're going to get when we split water is going to be oxygen. So we know oxygen is being produced, but it's not something that is being picked up, right, in data analysis. There appears to be no oxygen available. And so what would make sense then is that oxygen, because it has to still be produced, is also going to be used up somewhere in that process. So A says the oxygen produced in photosynthesis is used in cellular respiration, and that would make sense because we know plants will do this anyway. And so the algae would do that also, or at least the chloroplasts obtained from the algae would do the same. Photosynthesis can be sustained in the absence of oxygen production, and that is certainly not true because, as I said, in that very first step of the light-dependent reactions, when we split water, oxygen's produced. That can't be prevented. That's just a byproduct that comes out right at the beginning. So no, photosynthesis cannot happen if we're not producing oxygen. C, the plastids store oxygen for use in photosynthesis, and plastids, by the way, is a term for chloroplasts. Okay, different variation, different pigmented chloroplasts. Okay, so in this case, no, they do not store. We know chloroplasts don't store oxygen and gases in that way, so that doesn't work. And then D, the ability to carry out photosynthesis is transferred from the chloroplast to the mitochondria, and there's no indication that that's happening, right? Because the chloroplasts we know are present. So we're not just transferring the functionality of something. That doesn't happen. So your best answer option with this is going to be A. Number 10, based on the model of the life cycle of the sea slug, at which stage is the organism most likely capable of carrying out photosynthesis? And so I'm going to scroll up actually for this one so we can see this part. So if we're looking at this, notice there's a few things to kind of, that to me at least stand out. So I have the eggs early on in development. I have the larva, which notice the word they're using non-pigmented, which means it doesn't have that chlorophyll that we would normally see in chloroplast. Also non-pigmented here, mature to adult, and then we get a pigmented adult. So at some point between this, the juvenile and the adult stage, that these sea slugs are obtaining algae and then, starting to use their chloroplasts there. And so when we think about that, your best answer option when we're actually being able to do photosynthesis is when we have an individual that has those pigments, and that would be S, as a pigmented adult, okay? Continuing on in the same story, right? We have more about the specific genes that are found in the algae and then also found in the sea slug, okay? And again, if you haven't read this, I would recommend reading that. 11 says, which claim is most consistent with the evidence that the PBSO DNA sequences are detected in the eggs of the sea slug? And so what we want to see for that, or what we want to kind of account for in this case, the fact that we're seeing this DNA in the eggs of these sea slugs now tells us that it must have been there kind of to begin with, and it's probably being conserved in that region for a specific reason. And so in that case, what we think about, because it's in the eggs and is being passed then from parents to offspring, it must have been present in an ancestor in the past. All right, and so the best answer option for this one's going to be B, because it was present in a recent ancestor, and that then got passed down because it became beneficial for the sea slug group. It's not going to independently evolve because it's just too similar to the one that we see in the algae. It's not present in all organisms belonging to that phylogenetic clade. We just don't have enough information to make that assumption. And then this, again, because it's in the eggs for part A, or answer option A, it doesn't work because again, it's in the egg cells. And if it's in the egg cells, that means that the eggs haven't consumed anything. The parents had something, right, and then passed it down. So it's truly like one of those germline heritable mutations that we talked about with the very first question. Question 12, which statement correctly used the data from the table to support a claim that nuclear genes support chloroplast function in the algae. And so for this one, if we're looking for nuclear genes specifically, that refers to genes and DNA found in the nucleus, not in other parts of the cell. We know that in eukaryotes, chloroplasts have their own DNA. We know that in eukaryotes, mitochondria happen to have their own DNA. And that's going to be very specific and very different in terms of the genes that those organelles carry. But if we're going to claim that it's in the nucleus, well, we better find it in the nucleus. And so M is your best answer option. So the DNA sequences for the PBSO gene are detected in the nucleus, not in the chloroplast. If we find it anywhere else, like P says in the cytoplasm, that one's not going to work because that doesn't give us an indication that the genes are actually present in the nucleus. For R, just because they're found in many photosynthetic organisms doesn't answer the question about it being found in the nucleus, okay? And then S, those sequences are very similar. Again, that doesn't tell us location in the cell. That simply just tells us, the common ancestry, which is kind of what we answered here for number 11. So again, answer is M in this case. All right, 13, another carbon cycle diagram. So in this case, which action could lead to a reduction in atmospheric carbon? So if you use this diagram and look at it, you want to figure out the one, again, that's going to lead to a decrease in the amount of CO2 up in the atmosphere. And you can see we have all sorts of things: respiration, combustion, decay, and death, and root respiration, and animal respiration, that are all pushing more up in the atmosphere. The only one that's pulling out from the atmosphere is going to be photosynthesis. So if we have an increase in photosynthesis, then we're going to have a decrease in CO2 in the atmosphere. And that's why our answer is D. Humans planting trees in a forest, because then we have more producers that are photosynthetic, and theoretically then that's going to reduce the CO2 up in the atmosphere. Okay. The rest of these, powering a factory through combustion, burning logs in a fireplace also through combustion, and then eating mushrooms in a field, that's respiration, as we break down that food and create ATP energy. So A, B, and C actually all increase CO2 in the atmosphere. All right, 14. You've got a model of a bee pollinating a flower, and the question's going to be, what ecosystem services are best represented by this model? And this is truly something that is kind of a super subjective and difficult type of question, because we really didn't spend time talking about this year. This is one of those things that's kind of part of a standard, but is often not really discussed. There's one page in your textbook that goes through this. So when we talk about ecosystem services, we're talking about things that the natural world does to benefit us. All right, so if we look at these five answer options, and we want to choose three of them, we want to pick whichever three relate most to the scenario and also benefit humans in some regard, in a positive way, right? So M is going to be your first correct answer. That's called provisioning services, because here we are benefiting in that we are able to obtain and use goods produced or provided by these organisms. So again, it mentions that we get honey out of this process, out of bees pollinating flowers and going back to their hives and producing honey as kind of a byproduct of that. behavior. So certainly we get that as a positive type of benefit here. P is also going to be a benefit. So P is the second correct answer. Benefits obtained from regulation of ecosystem services. So these are called regulating services. And what this means, we're talking about the regulation of an ecosystem in a manner that allows pollination to continue happening. So in other words, providing lots of biodiversity and ensuring that there is enough biodiversity to make sure that we don't have to deal with things like pests that could destroy these crops of flowers that then in turn could impact the bees' behavior, right? So as long as there's lots of biodiversity in an ecosystem, there are things that will eat herbivores that like to, chew on plants and could potentially harm them. So those regulating services are also represented in this model. The last one is called supporting services, which is S. And this is, again, services necessary for production of other ecosystem services. So in other words, they're all kind of interconnected in this case, right? And all of those provisioning, regulating, supporting are all going to be kind of functioning together in a way to benefit humans. Not really that's the point here, right? But in terms of this question, that's what they're getting at. The two that don't work, cultural services are and land use. If we have land use, clearing land for human housing development, that is not a benefit to us. That's also not a benefit to bees. And when I say it's not a benefit to us, I mean in terms of our food supply, right? I mean, it may provide housing and those types of maybe short-term benefits, but clearing land can also have a super negative impact on pollinators and therefore our food supply. Cultural services, non-material benefits from ecosystems, that's not something that we see here. That would be more of like, conservation type things, protection of a national park, and then the ability of tourists to go and see these things that they wouldn't be able to see without that protective feature in place. Okay. So again, answers here, M, P, and S. 15, we have succession. And again, I mentioned this before. We had primary and secondary. And in this case, if you notice, we're starting with bare rock. We're going to end with what looks like mature trees, and we have an increase in soil depth. So the question is, which two natural events would lead to this type of succession? And when we look at this process happening, we have to have something that really clears out an ecosystem in a way that there's nothing going to be living there. Okay, and so when we think about this type of process, there's one thing that I usually tend to focus on, which is going to be volcanoes, and another one, which is glaciers. So the two answer options that are going to be your best are C and E, which deal with volcanoes and glaciers, because both of those result in really the production of an ecosystem that is brand new. If you have a lava flow that cools and hardens and forms igneous rock, I mean, there's nothing living beneath that surface that's going to like creep up through that rock in that way, right? We're talking about, all right, now we have to go in and recreate new soil, and we're essentially starting with bare rock. Same with the retreat of a glacier, right? Glacier are these heavy, thick, and dense ice sheets, and they move very slowly across the land. And as part of that process, because of their depth and their thickness and their weight, they simply kind of scrape everything beneath them. So anything in the path of a glacier, though it's very slow moving, is going to be scraped off in a way to where it just can't exist when the glacier moves. Like it's just going to be soil and bare rock, and that's it. Okay, clear cutting of forest, wind damage, and flooding, all of that leaves things there. Even though it may destroy some populations, there are things that remain, and so growth will start from wherever it left off. All right, so 16, we're looking at shark catch rate over time and a shark population in this ecosystem called Shark Bay. And so the question is, which claim about the population is best supported using the data? And this one I think is a little bit tricky. So M says more people fish for sharks in the winter than in the summer. And that would be something that we can just, first of all, we're looking at the catch rate. So the assumption is that in other words, we're not looking at the number of people out fishing, we're looking at how many are actually captured, right? Now, obviously, based on this, my lighter gray bars are a whole lot smaller and maybe even non-existent for some years, which would maybe point to this idea that there's probably not as many people fishing for sharks in the winter than in the summer. So it's likely that M doesn't work because it's probably the opposite. It's probably more people fishing in the summer than the winter. But again, we don't have that measure here either. So we also can't just make that assumption. P, sharks have larger body sizes during the summer than during the winter. And that one doesn't work either because we just don't know. It has nothing to do with, in this data that they're providing, the mass of the animals that are caught. So we can't make that claim, and that can't be a correct answer. R, sharks migrate northward from Shark Bay during the summer to maintain a low body temperature. And perhaps that's the case. During the summer, in that case, we would be looking at, summer into winter time, right? And that's why maybe that there are less sharks there present at that time. But again, we don't know that, and that's harder to support with this data. S, the population of sharks in Shark Bay is larger in the summer than in the winter, and that kind of makes sense because they're catching a whole lot more in the summer as compared to the winter. And so S is actually your best answer option given those different choices there, okay? Because the other ones simply aren't supported by the data that's provided. All right, so again, S for number 16. 17, which 3 processes occur during meiosis? And for this one, you want to think back to chapter 12, because that's where we talked about 12.4, I believe. So during meiosis, we talked about crossing over as occurring in prophase 1. That will be one of your 3 processes here. Meiosis, remember, is only used for sexual reproduction, not asexual, so B does not work. Formation of gametes, certainly we do gametogenesis as meiosis concludes, and that includes the formation of sperm and eggs. Independent assortment also occurs during meiosis, so D is also a correct answer, and that's where the homologous pairs separate at random and independently of one another. Okay, and then again, that would be an anaphase 1 of meiosis 1. E, answer option E, says production of diploid cells, and that one does not work because during meiosis, our goal is to create gametes that are actually haploid and have half the genetic information of the original parent. Diploid, if you remember, has 2 sets of chromosomes, or the whole coming from their original parents, the 2 parents that together their gametes join to create that individual. So diploid cells are produced through mitosis. Haploid cells are produced through meiosis. So your answers here, A, C, and E. 18, we have a type of membrane receptor called RTKs, which do this really interesting thing along with many other molecules in our membrane, many other receptors like this. They do this process called enzymatic phosphorylation, and that's what you see right here. And I know that is a mouthful. Enzymatic phosphorylation. What that essentially means is that when they interact with a signal, which we're seeing here in this diagram, signaling molecule comes in, it's going to activate this receptor, and this is our receptor. Here's the inside of my cell, and here's the outside. So my signal is going to bind to the outside of this protein that's stuck in the membrane. It's going to activate the protein, and as a response to that, it's going to add a bunch of phosphates. So when we say enzymatically phosphorylate itself, it's adding phosphate molecules to the protein, and as a result of that, that initiates a response in the cell, okay? Some of these words, again, are going to be the trickiest part of your test. So pull out the parts that you've heard of. We've talked about enzymes. We've talked about a phosphate group. So see if you can't, derive meaning from these terms if you see them and they look a little bit overwhelming. So your question here is, a researcher is planning an experiment to engineer a non-functional version of an RTK. Which engineered protein will most likely be non-functional? Excuse me. So if we want to talk about taking a protein and making it inactive, non-functional, that usually means we need to change its structure in some way or change the conditions in which it's found. So if we look at M, that is changing the structure. M says we're going to have a second enzyme region in the cytosolic portion, which is going to be here in the cytoplasm. Cytosolic means cytoplasm. So inside the cell, they're going to say we're going to add another enzyme region. That likely won't do anything, though, because it appears, based on this, that my signaling molecule can only interact with what's on the outside of the cell. It probably cannot cross the membrane. It's probably polar, hydrophilic, charged, or too large, okay? So this one probably won't do anything. Answer option P says an RTK protein that can phosphorylate itself even in the absence of a signaling molecule. That's likely going to kind of cause it to become fully active all the time. right? It doesn't need a signal. It's just going to be active, 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 because it's going to phosphorylate anytime it wants. So in that case, it's going to do the opposite here of making non-functional. It's going to be more functional. R, an RTK protein that is unable to bind to the signaling molecule. Well, that would make sense, because if we can't bind up here, then we can't activate this protein, right? So that phosphorylation process doesn't happen. The cellular response also doesn't happen. In S, an RTK protein that has multiple phosphorylation sites, that probably won't do anything either, even though it does involve changing the structure, because it will still likely initiate the same response. And if anything, it may initiate a stronger response, which is the opposite again of not functioning. So your best answer option is R in this case. 19, a removal of a dam in a river has restored river flow and improved the water quality of the river. Which of these would most directly increase due to the enhanced water quality in the river ecosystem? My words are losing me. So when we think about this, if we're going to enhance the water quality, that means we're going to make it better, right? And so we're going to improve it, and that means better things for the things that live there, right? More survival, probably larger population sizes, and so on. So if we're going to increase that, chances are we're not going to get more pollution, because those are kind of contradictory things, right? Anything else that tends to be negative is also not likely going to happen. And so death rates are negative, right? Which would imply that more species are dying. Global climate change also would likely have no impact on this scenario. And even if it did, it wouldn't have a positive effect. So most directly increased in enhanced water quality is the increase in biodiversity, or B, because again, the more positive these things are, the more nutrients available, the more space, the better the quality of water in this case, the more things can live and survive in that place. All right, on to number 20. So we have, again, another series of questions. So this is about cyanide, right? And again, I'm not going to read through this, but make sure that you do. Essentially, what we've got is we're looking at the inner membrane of the mitochondrion, where we are doing the electron transport chain. And it says, excuse me, that the protein that's in the membrane here, in the absence of cyanide, does a specific job, and that it's going to push electrons along, right? If cyanide is present, then we can run into some issues to where the mitochondrion can't use oxygen properly. So like this component right here, based on that, the fact that I see CO2 right here, this component here must be impacted. So question 20 says, based on what we see in that inner membrane, which model correctly represents the effect of cyanide on cellular respiration in herbivores that eat clovers? So if we're going to produce cyanide, and we're going to have this where oxygen is not going to be used, well, the downside of that is I don't really have a place where I can push some of these things that were used as part of this cellular respiration process, this electron transport chain. And if I don't have oxygen, I know I can't form water, right? And if I don't have the ability to form water, I don't really have anywhere for my electrons to end up going. And I also don't have the ability to drive this process here through my ATP synthase. And so the production of ATP is also going to be impaired. So your best answer option for this is going to be P, because the impacts that we're seeing, the inability to produce water, inability to produce ATP. M and S both say we're not going to produce CO2. And if you look back at the diagram, that's not even part of this, right? So there's not going to be an impact there. It's going to be simply the production of water and ATP, two things that are shown in that diagram. All right, so it kind of goes to show you, even if you read these questions and it seems overwhelming, you can work your way through these things. The diagrams will certainly help. They're providing them for a reason, okay? 21 says, which statement best describes how sugars are metabolized in herbivores that eat clover containing high levels of cyanide? And so we know that we're going to have an issue here with that electron transport chain, which is one of the aerobic parts of respiration. It's the very last step, and it's a part that normally makes a ton of ATP. So when we think about this, we know cyanide, as it mentioned in the scenario there at the top, is toxic. So A says we can use these alternative processes. We can use fermentation and Krebs cycle to break down or metabolize sugars, that means break down, and produce sufficient ATP to sustain life. In other words, enough ATP to keep that living thing alive. But cyanide is toxic, and so we know that keeping them alive is not what's going to be happening here. So A does not work. In addition to that, Krebs cycle is aerobic, and so if we're not able to use oxygen properly, we're not doing the Krebs cycle. B says aerobic respiration photosynthesis partially metabolized sugars, and I don't even have to read the rest to know photosynthesis does not break down sugars. It does the opposite. And we're also talking about herbivores. So herbivores aren't doing photosynthesis anyway, right? They don't have the ability to produce their own energy through that process. So B doesn't work. C, glycolysis metabolizes sugar more effectively than it does in the absence of cyanide, produces a normal amount of ATP per sugar molecule, which is very much in line with answer option A, except that it's referencing just glycolysis. And glycolysis, we know, cannot change how many sugars it makes. We always get how many ATPs we make. We always get 2 ATP, a net gain of 2 ATP out of that process. And so that simply doesn't work either. Best answer option, the correct answer is going to be D. Glycolysis and fermentation metabolize sugars but do not produce sufficient ATP to sustain life. So in other words, these two anaerobic processes will happen, but it's not going to be enough to allow those life functions to continue. 22, which statement describes a likely metabolic impact of cyanide production on clover plants? Well, what's interesting about this is that cyanide is also toxic to the plants that are producing it, but they do it as like this last-ditch effort to kind of evade herbivores and really protect the rest of the group. So your answer for this one is going to be P. Cyanide is toxic to clover because they use, that same enzyme for their ETC in the mitochondria. Very similar to what we see in animals because remember, plants also do that process. So the issues with some of these other statements is that they kind of reference these things in the wrong manner. So like M says cyanide is toxic because they produce ATP without the ETC. That's not possible. They can do glycolysis, but they're not going to do that in its, when they have the ability to do the electron transport chain, right? As long as there's oxygen available, all organisms default to using Krebs and ETC to make ATP. R says cyanide is toxic because they use the enzyme for ATP synthesis in the chloroplast, and that doesn't happen in the chloroplast. If it had said mitochondria, that would be a whole lot better. S, cyanide is non-toxic because they carry out electron transport in chloroplasts. Again, they don't do that in the chloroplast, they do that in their mitochondria, just like we do. So again, let me repeat one more time. P is your answer for that one. Same kind of setup here. We're looking at part 2, and we're looking at the presence or the absence of these two genes. And so for this, we've actually got kind of a complicated question. It says clover plants that are heterozygous for both genes, so both the A and the L gene, if they're heterozygous for both, meaning big A little a and big L little l, if they're crossed with clover plants that are homozygous recessive for both genes, and it says that they're going to produce 300 seeds, how many would actually produce cyanide? So to figure that one out, we have to know what results in the production of cyanide. So if we're going to have the production of that, we look at this very last sentence here. Plants must have dominant alleles of both genes. They must have at least one big A and one big L to make cyanide. So this I'm going to set up actually like a dihybrid. I'm going to have big A, big L, little l from one parent, and then little a, little l, and I'm going to cross those to figure out how many offspring actually have that genotype and then phenotype. And again, I'm looking for individuals that have both one big A and at least one big L. Now, the way that you set it up, I'm going to hold up my answers, and I know my screen is kind of small here. Let me scoot it over a little bit. So this is going to be your parent cross. So we end up with big A, big L, little l, cross those together. Those are my gametes that I get out of that. All right, and if I go through that cross, I know a few things about it without actually, myself having to set it up. But I'm going to end up with 25% that are going to be big A, big L, little l. 25% are going to be big A, little l. And then a remaining, 25% are going to be little a, big L, little l, and then little a, little l. So really I've got 1/4 probability for each individual genotype there. Now, my phenotypes are going to be different in this case, but the only ones I'm concerned with are the ones that have at least one of each, at least one big A and at least one big L, and that's 25% of the offspring from that cross. Now, they give me a number here. Grand total is 300. And so in this case, 25% of 300 is going to be 75. All right. And if you didn't know that, that's some math, right? Divide 300 by 4 or figure out, what would be 25% of that number. Or you can go the opposite way and say, okay, if I add 75 together 4 times, do I get 300? And your answer would be yes. I mean, and really the other answer options are kind of, as long as you know it's 25%, the other ones don't work. 0 out of 300 is not anywhere near that. 150 is actually 50%, and 225 would be 75%. So your answer here is B, 75. In a controlled study, we've got 2 parental plants. We produce 240. All the F1 plants produce a cyanide, but only half produce linamorase. So if we go back, linamorase is coded for by that L gene, right? In this case, what are the most likely genotypes of the parents? Well, we need to have all of them having at least one big A. And we need to have a few that are going to have the little L's, right? Because it's got, sorry, I had to think about that. It's got half of them that are going to produce the linamorase, which means half of them have big L and little l, or big L, and the other half are going to be little. So here's the cross for this one. All right, and so this is what it would kind of set up as being. So if we had answer option S down there, which is our correct answer, in that case, we're crossing big A, big L, little l, with big A, little L, little l. And those are going to be the potential phenotypes and genotypes that I get out of that, okay? So that's your best answer option. I guess I misspoke. S is big A, big L, little l, and then big A, little l, little l. Okay. So that should be the correct one. And if you set up these crosses, by default, if you see a question like this and you're not sure, it doesn't hurt to set them up if you have time and actually cross them, right? I mean, that can be done if you need to kind of check. 25, you've got a series of pedigrees. It says each symbol represents an individual clover plant. If the shaded symbols represent plants that produce an enzyme, which pedigree accurately represents the inheritance in clover plants? So what we're trying to identify here, given this, we know that linamorase is going to be, the production of that is a dominant type of trait, right? And so we don't have the ability to see it appearing in the parents, having it skip, and then having it reappear down here. And so because it's dominant, that's what you want to look for in this case. That was one of our look fors when we talked about pedigrees anyway. For a dominant trait, it has to appear in every generation, at least between those that are affected and then others that are affected down here. Each generation in between should also have it. So if we look at answer option A here, these guys don't have it and these guys don't have it, but their offspring do. So this one doesn't work. Okay, if we scoot down to B, kind of same thing here, right? We don't have it in the original parents, but it appears in 2 of their 4 offspring, and then it appears in some of the grandchildren or grand plants, if you want to call them that, after 2 generations. It comes out of nowhere. And that, again, can't happen with the dominant trait. This one looks good. It's in every generation, except for we have this weird scenario right here where it's not being passed down. But, I mean, that's reasonable, right? I mean, we're seeing it appear in at least 50% of every generation. It was in 3 of 4 of the offspring here, and even more than that over here. And then if we consider D, this one also doesn't work because, again, we're skipping, right? We're skipping over here in this kind of subset of the family and also this one here. So your best answer, the only one that works in this case, is going to be C. All right, 26, an intestinal cell is shown. Cells that line the intestines break down food and absorb nutrients. Which organelle performs a similar function for cells? So we want to know what's going to break down food and absorb nutrients. And so the answer for this is going to be the lysosome. The nucleus, remember, stores information. The Golgi apparatus helps to modify, sort, package proteins. It's like the UPS or FedEx of the cell. The mitochondrion helps to do cellular respiration, which, may make you think, okay, breaks down food. That could be something that's done there. But the difference is that the lysosome can break down all sorts of different types of food molecules. The mitochondrion is really specialized for just breaking down glucose and doing cellular respiration. So lyse, if you remember, stands for to split, loosen, or break. And when I see the word breakdown, that's usually where my mind goes. Okay. And so hopefully that can be maybe helpful to you also. Last question for this first half, 27, which two viral characteristics provide evidence to support the argument that viruses are more similar to living components than to non-living components of an ecosystem? So in other words, what is it that viruses have that makes us think that they could potentially be categorized as living things? And so we know there are some general characteristics of living things that everything has to have. And one of them is that they have to have cells, but viruses don't have cells. So B doesn't work. Even though that's a characteristic of living things, viruses don't have cells. They're just genetic material and proteins. That being said, A works, right? They do contain genetic material, which we know all living things also have to have, DNA or RNA. Viruses cannot store energy to grow. They can't really even do that on their own, on their own. And they don't have the ability to do fermentation or glycolysis or Krebs or respiration or photosynthesis or any of that on their own. So C does not work. They also cannot reproduce independently. They rely upon host cells for that process. And so they need to use the machinery in a cell that they're going to kind of attack and invade in order to produce more viruses. They cannot do it on their own. So D doesn't work. And then E, the very last one, they can evolve through mutation. They can evolve through natural selection. And we see that right now with COVID-19, right? I mean, there are new mutations, new what they call variants in different parts of the world that are kind of spreading and are making it a little bit harder to try and deal with that viral pandemic. Okay. So those are the characteristics that we think of, again, in talking about living things. And again, the ones that viruses have, DNA or RNA, and the ability to change over time, okay? And fairly rapidly, they can do that. So very long screencast for this. Again, if you hung with me to the end, amazing, thank you. If not, no worries. I will do another one here for the second half of the EOC practice test. So again, if you got any questions, let me know. Otherwise, I may or may not see you next time. Bye, y'all.